cell cycle. The cell cycle is divided into two stages. The interphase, in which the cell is growing and preparing for mitosis division, and the mitotic phase, in which the cell is dividing and the nucleus is dividing. We are not concerned with the interphase stages right now. You know them. The first gap, the S phase where DNA synthesis happens, and the second gap. What you are required to know, though, that mitosis lies between the second and the first gap, after gap 2, G2, and before G1 of the next cycle. The mitosis, as you know, it is divided into 5 subphases. We're going to talk about the main 4 today. Prophase, followed by metaphase, anaphase, and then telophase. Once again, mitosis is where division happens. After mitosis, after the nuclei divide, you have to separate the cytoplasm of the two cells, a process called cytokinesis, which differs between the animal cells and the plant cells, as we're going to see today. As you can see, most of the time in the cell cycles, 90% of the time of the cell cycle is in the interphase stage, which is the stage of growth and function and the functionality of the cell. Same, the first gap, the S phase, and the second gap, followed by mitosis. Let's start. This is a light microscope for the 4 main stages of mitosis. What you can see, you know what happens during prophase. The nuclear membrane degenerates, start to formation of mitotic spindle, etc., etc. But what we are concerned here is what you can see under the light microscope. You always judge by the nuclear material. In the prophase, the chromatin starts to condense to form chromosomes. Okay, you start to see the condensation of the chromatin. Followed by the metaphase. As you know, the chromosome align themselves in the midline of the cell in an imaginary plane called the metaphyseal plate. These threads are the tubules of the mitotic spindles, microtubules synthesized by the centrosomes on the poles of the cell. After that, we have the anaphase, separation between the 2 sister chromatids of each chromosome, and they start to move away from each other. Finally, you have the telophase. Each set of chromosomes will align themselves at the different poles of the cells. In the prophase, we started to see condensation, while in the telophase, you're going to start to see decondensation. The chromosomes will form back into chromatin mass once again. After the telophase overlaps with it, we have to separate the cytoplasm of the cell, as you can see here, the formation of a cleavage furrow during cytokinesis. Of course, these are all animal cells, right? They are rounded cells with irregular cell membranes all around. Next slide. Once again, you judge always by the nuclear material. Here you see the nuclear material is condensed, the genetic material is condensed, the chromatin is condensed to form chromosomes على طول. This is a prophase. Now, is this an animal or a plant cell? How did you know? This is a plant cell, that's correct. How did you know that? Yeah, exactly. It's quadrangular in shape, it's polyhedral. As you know, the plant cells, they have rigid cell walls that give them the regular shape. This section was taken from the root of a plant, the ependyme of a root. This is zooming out in the lower slide into the nucleus during the prophase stage. Here we compare. شو prophase متى ولا يا شباب؟ شو رأيكم؟ How did you know that this is a prophase? Always by the genetic material, condensation of the genetic material into chromosomes, no mitotic spindle apparent, no metaphyseal plate, no separation. This is the prophase stage. Here we compare between an animal cell and the plant cell. We can determine what type it is, as we said, looking at the cell membrane, the walls of the cells. This is an animal cell, rounded, irregular plasma membrane, while the plant cells, they have rigid cell walls. Here, the metaphase. For an animal cells, the chromosomes are aligned in the metaphyseal plate, an imaginary plane in the center of the cell. And metaphase in a plant cell, what you have to know is that the numbers of chromosomes differ between the 2 types of cells. As you can see, the color is denser in the animal cells due to the larger number of chromosomes, which makes sense. The animal cells has more multiple enzymes, more multiple pathways, thus a larger amount of genetic material. While in the plant cell, you can see the chromosomes clearly separated due to the reduction of number of chromosomes in the plant cells. The anaphase stage in animal and plant, clear the separation between the two sister chromatids. And once you see the two masses of chromatin massed together, two different masses at each pole, you know that you are in the telophase. You see there's no, the appearance of chromosomes has disappeared. No sticks, ما في عصي, ما في هون, لأن شو حكينا هون بصير decondensation of the genetic material from chromosomes back to chromatin once again. Overlapped with the telophase is the cytokinesis, which is a major difference between the animal cell and the plant cell. The cytokinesis in the animal cells is by formation of the contractile ring, the cleavage forum. While in the plant cell, there will be formed a cell plate, a vertical cell plate that will separate the two newly formed cells. Animal or plant? Plant, easily. What stage are we? In what stage are we? Telophase, right? Two masses in different poles of the cell. Do we have cytokinesis in this section? Yeah, here it is. We have a cell plate forming in the middle of the cell to separate the two new sets of chromosomes. This photo for an animal cells in the oldest stages. The first one, prophase. The second one, metaphase. After that, anaphase, and finally, telophase stage. Once again, these are plant cells. What do we have in here? What stage are we in? Prophase. What about the other cells? As you can see in the other cells, the nuclei are paired, which means that these are non-dividing cells yet. These cells are in the interphase stage. They are still functional and preparing for mitosis. What about this? Animal or plant cells? Animal cells, yeah. Irregular shape. In what stage are we in? Also in the prophase. Okay, once again. كان شغال مش شغال كان ما شفتيش والله حدا عنده صار هيك مشكلة كمان also prophase in an animal cell once again repetitive plant cells here. What about this one? What stage? Metaphase. What about this one? Prophase, right? And the other cells, interphase, non-dividing. They are still pale nucleus, pale chromatin, non-dividing cells in the interphase stage. Animal cells, clearly in metaphase, the cells are aligned in the middle. Plant cells, this one, what stage? Anaphase, this. And this also, prophase. While the others are still interphase, but probably these two are preparing, right? The color is getting denser, but not yet. We are not in the prophase yet. You cannot see separated chromosomes yet, so you did not enter the prophase. Anaphase, that's right, in an animal cell. Here, what about here? Ana or telo? Telophase. They are not only separated, they start to form masses again. They are starting to decondensate once again. This is a telophase in a plant cell. Can we see a cell plate in here? Not yet. Maybe cytokinesis did not start yet, but as you know, cytokinesis always overlap telophase, but not right at this moment, not yet. It will be followed by the formation of cell plate. Here, of what? Animal or plant? Did cytokinesis start? Yes. What do we have in the middle? What do we call this? This range? The cleavage forum in an animal cell. Different sections, light microscope for plant cells. All the number ones are what? What stage? Prophases. Number two, what about that? Metaphases. 3, anaphase. 4, telophase. And in this 4 here we can see the cell plate. 5, it's a cell in the interphase. After division, it's functional, it's the interphase stage. Here's again the same thing. I guess you can all identify the different stages. Here the telophase is very clear, and the decondensation of the chromosome back into chromatin mass is also clear. Finally, we're gonna, I guess, أخذته بالtheory, I guess, أخذته بالtheory. في different subphases. We have a pro-metaphase, early prophase, late prophase. You are not required to identify these phases under the microscope. In the practical, you are only required to recognize the 4 main stages: pro, meta, ana, and tero. That's it. Study راجعوا الصور إذا بدكم، إذا حد عنده سؤال يناديني.